And hello there, microbiology friends. So we're going to pick up on our respiratory diseases, starting first with upper respiratory diseases. Behind me I have some incredibly disgusting pictures. Here's hoping I don't have to say the word anus today. Oh, rats. I just said it. Anyway, what we're going to do is go through some examples of the different kinds of bacteria and the different kinds of effects that happen when bacteria get into the upper respiratory tract. So you'll know some of these diseases. Hopefully you'll never get, and maybe you've never heard of, and we'll see where we go from there. That's something that all of you know of is the effects of streptococcus, specifically strep pyrogenes, which causes the common strep throat. So it's gram-positive. It is a streptococcus in terms of the way it looks, these little beads, and it's got a capsule. And part of what makes it so damaging is that it does have a variety of toxins, exotoxins that it can release. I mean, that's one of the things we're going to see with these bacteria is that pretty much the bacteria itself isn't all that damaging. It's going to be the candy coating, the exotoxins it releases, all kinds of stuff like that is really the damaging part of it. So a couple of things that strep has been involved in, of course, is strep throat, which you probably are all quite familiar with. Necrotizing fasciitis, damn, I don't want that. Flesh eating. Toxic shock syndrome is another real serious problem in which you have a large enough bacteria load that begins to secrete the exotoxins, and that itself becomes toxic, becomes deadly. And so you can have a rather benign bacterial infection, but the fact that it's dumping out all those exotoxins makes it really quite deadly. Scarlet fever is one of these things that, as a cell biologist, it doesn't do particularly a lot of human pathological work. I'm always sort of, questioning the different kinds of terms and these old-fashioned terms. So scarlet fever is the result of a strep infection. And so what can happen is it is essentially a whole range of immunological effects that occur after a strep infection happens. And it can cause a whole range of conditions, including blindness and deafness. Potentially, it's what blinded and deafened Helen Keller. Anyway, so that's strep. And, initially it is getting in through the lungs and through the oral cavity. Diphtheria, or this is a Corynebacterium diphtheriae. This causes diphtheria, and a couple of interesting facts here. It is a gram-positive. It's got sort of a club shape to it. It actually has a little bit of a head and then a narrowing tail. It has a number of different kinds of pathologies in that it can be in the throat, laryngeal, pharyngeal, getting further down. It also can be into the tonsils, in the anterior nasal passages and things. One of the classic symptoms is something that's referred to as a bull neck, which you see here. It is a increasing size of lymph nodes, and this is part of what, of course, the tonsils are, and they were responding to. Often you have a deep cough, and the exotoxin actually comes from the fact that this bacterium that is pathogenic has incorporated a phage. It's something that's called a lysogeny event, in which the bacteria virus has actually incorporated itself into the genome. So it can cause a variety of different kinds of damages. If left untreated, it can become systemic and get into the heart and also in the nerves. Pretty bad news. So there were millions of cases per year prior to the vaccine. So you know that you've taken the DPT shot, diphtheria, pertussis, and tetanus, I think. Anyway, diphtheria, we have a very good diphtheria vaccine. And so now after the vaccine, we've gone from millions of cases to a few thousand per year, mostly in Africa and India and Indonesia. In the U.S., there's virtually no diphtheria. So that's a good +1 for vaccines. Now, just as a little note, we talk about exotoxins. Exotoxins can do a variety of different kinds of things in the manner in which they interact with the cell. In this specific case, we have an exotoxin that is known as the AB, very creative name, and the AB toxin actually can interact with a receptor, a normally occurring receptor on cells. And this is an epidermal growth factor-like receptor. Not that it's important. It's the fact that that's how these things are getting into the cell, is by binding to a receptor, and then through a phenomenon known as receptor-mediated endocytosis. It is essentially the cell's response when a receptor binds its binding partner, what's known as a ligand. That binding changes the shape of the receptor up there, and it causes that part of the membrane to get sucked in and form a bubble, form a little membrane ball or a vesicle. And with this vesicle then is brought into the cell, and there are a whole range of changes that happen into this vesicle. A typical thing that happens with receptor-mediated endocytosis is that hydrogen ions, protons, are pumped into the vesicle, dropping the pH. That begins to slightly denature the proteins, and the goal is, under normal receptor-mediated endocytosis, you can knock the hormone, or whatever it was that you brought in, off of the receptor, and the receptor can either be recycled, or the whole thing is actually just broken down into amino acids and just reincorporated back into the cell. Regardless, this toxin responds to the increase in hydrogen ions, or a decrease in pH, and it releases a portion of the exotoxin, and this portion actually is involved in binding to something called the elongation factor, which is a critical protein that's involved in protein synthesis. It's actually one of the proteins that is involved in ushering tRNAs into the ribosome and allowing for the messenger RNA to be read, and for the ribosome to move over 3 nucleotides at a time, the codon chunk. And it is so potent that one toxin molecule can kill an entire cell. So this is pretty damn serious. So these toxins then can be real serious problems. And this is the kind of stuff that is involved, what we think of as a virulence factor. So if you don't have this toxin, you can be a diphtheria bacteria that's not particularly toxic. All right. Other kinds of bacterial diseases that can get in through the respiratory system. We have a range of bacteria that can cause a generic kind of disease called bacterial meningitis. Meningitis is an inflammation of the lining, the meninges of the brain. And obviously this is going to be a real serious problem. And so getting bacterial infections in your noggin, that's not good. So the route is through the sinuses, the head, the throat. You can also get it in through the blood, so through different kinds of wounds. Neisseria meningitis, meningitis. It's gram-negative. It's aerobic, and it's non-motile. It doesn't move around very well. It's one of the bacteria then that is able to generate a inflammation in the brain, and this is a specific problem primarily for teenagers and young adults. Happily, we have a vaccine, and probably you have all been vaccinated against it. So, one of the things that's not all that common, but it is quite deadly. So having a vaccine for it is really nice. Other bacteria that can get in through the upper respiratory tract, Streptococcus pneumoniae. When people talk about having pneumonia, what this is they're describing the condition in the lungs. The causative agent is often strep pneumoniae. And also, you can get what's known as viral pneumonia, which is a virus that is affecting the lungs. And so you end up with having fluid accumulating in the lungs. So the term pneumonia is a description of the symptoms and the congestion you have in your lungs. The causative agent can be a variety of things, but strep pneumoniae is one of the primary causes of pneumonia. There's a vaccine to it as well. One of the points is that you're going to have strep pneumoniae in a lung culture. You know, we all have strep growing all over us, a variety of different kinds of streptococci. And so the presence of strep pneumoniae is not in and of itself indication of having, quote, pneumonia, because we all have it. It's only when your immune system becomes somewhat attenuated or kind of beat up that the bacteria can start to actually grow. So a classic thing that happens is when someone has a really bad flu caused by the influenza virus, that can sort of tamp down the immune system, make you a little bit more sensitive, and the bacteria that's in your body to begin with, but you normally keep it at bay, begins to grow and begins to replicate enough that you start to have symptoms. And so that's how you can get a secondary infection after the flu, is because of the immunosuppressant activity of the flu. This is another point. Folks that are on immunosuppressants, like after organ transplants, they're going to be very susceptible to all kinds of bacterial infections like this. So, we have these bacteria on us. It's just that our immune system is keeping them at bay fairly well. Here's another one of these guys. The name is a little confusing. Haemophilus influenzae. This is not something that causes the flu. This is a bacteria. The flu is caused by viruses. It's a gram-negative. It's an anaerobic bacteria. And happily, we also have the Hib vaccine, which blocks this bacteria from getting a foothold and expanding and causing lung problems as well. All right, so there are a range of other bacteria that cause upper respiratory infections. There are some pseudomonads. There's another Streptococcus pneumoniae. This is a strep that is causing pneumonia. Haemophilus influenzae, as we mentioned here. Let's see. We got a variety of different kinds of symptoms and organs that can be affected by these different bacteria. The epiglottis, that's the little bag in the back of your throat, it can become very swollen. Apparently it killed George Washington. I wasn't aware of that, but there you go. These bacteria are often responsible for middle ear infections. And if you've got a little kid, little babies get middle ear infections a lot. My general response, though, is having at least my child, he'd wake up in the middle of the night with a middle ear infection. The next morning we go and we get antibiotics, and we kind of decided that, if you just kind of wait around for a while, it usually fades back. And rather than giving your little ones excessive antibiotics, it's better to just hold off for a little while and see how it's going to go. That's my opinion anyway. Sinus infections are often caused by a variety of these bacteria. Tonsillitis, again, another bacterial infection. Pharyngitis and laryngitis. These are bacterial infections that hit the throat and a little bit lower. And they're all bacterial infections. So antibiotics will be our best course, but probably maintaining a good immune system is your best thing you can do. And for some of them, of course, we do have vaccines. So that's upper respiratory. So we're talking that would be sinuses and the mouth and throat. Now let's move into the lower respiratory tract. Probably a bit more serious, not as easy to get into, but now we're talking about the lungs and going to start to impede our oxygen carrying capacity. A disease that you know of caused by Bordetella pertussis is known as whooping cough, named because of the very loud coughing and inhalation that occurs, sort of a wheezing whoop, and the cough can last for a very long time. We prevent it with the same vaccine that we use to prevent diphtheria, and that is the DTaP or the DPT vaccine. And whooping cough now is really pretty darn rare. In the past 10 years or so, cases of whooping cough actually are starting to come back up a little bit, partly or in part because of failure to vaccinate. So again, whooping cough and the Bordetella bacteria, it causes the problem because of the exotoxin. In this case, we've got an exotoxin that is being bound by a receptor. The receptor then, through receptor-mediated endocytosis, pulls this portion of the membrane in. We end up with this vesicle, referred to as an endosome, that has protons being pumped into it. That causes the release of a portion of the exotoxin into the cell. And this is going to actually interact with a protein known as Rho that is involved in a whole range of different cellular effects. So we end up having the cytoskeleton being disorganized, which in turn affects the cell's ability to divide. It can also begin to affect the turning off and on of different genes. And finally, because of some of the variety of effects, it can block cells from progressing through the cell cycle. And, like neurons and the brain cells, they don't really divide much at all, but the lining of the lungs is constantly being sloughed off, and so you have to have a fair amount of cell division going on there. So it can end up having a variety of serious problems in the lungs in this way. Another bacteria that you perhaps have heard of is the bacteria that causes tuberculosis, Mycobacterium tuberculosis. This is an aerobic bacteria again. It is a rod shape. It's got actually a fairly weird cell wall. It's gram-positive, but it does have a strange cell wall. Not that that's all important. The symptoms are cough, and often a productive cough with blood, fever, malaise, dramatic weight loss, very high mortality rate if it's untreated. And there are antibiotics that can knock back tuberculosis bacteria, but you got to be on them for a long time, and we're beginning to see resistant forms of tuberculosis. In real dense populations in New York City, there are cases now of antibiotic-resistant tuberculosis, which is a bad deal. So, that's why we try to avoid the overuse of antibiotics, because, as we've been talking about, the use of antibiotics is going to begin selecting for those antibiotic-resistant mutations that are in the population. And so you end up with antibiotic resistance that then in turn cause or requires the use of more powerful antibiotics, and you kind of work your way up to the top to where you're using the most powerful antibiotic that we might have on hand and hope that you don't have resistance to that. Tuberculosis is manifests itself as these spots in the lungs that become electron or X-ray opaque. As you begin to see them in the X-rays, these tubercles, this is these congested areas that are beginning to fill with all kinds of different sort of inflammatory products in the lungs, and so it begins to inhibit the ability to breathe. A pretty serious disease. Now, we've got, let's see, what was it? Yeah, but look at this. All right, so tuberculosis, there are, the primary infection mechanism is through the lungs. So you can get exposed to it, and most folks aren't going to have a problem. But in 30%, we will end up with having lung infections and begin producing these TB lesions known as the tubercles. We have a range of different infections that occur as the bacteria begin to colonize these tubercles in the lungs. So we have the cavities in the bronchi, that is, in the side of the lungs, that are little canals, if you will, or tubes that then branch into very, very fine tubes, where you actually have oxygen exchange going on. These begin to get blocked up with the bacteria. And one of the things that starts to happen is that as the bacteria are replicating inside the lung, coughing is a way of expelling bacteria. And so the bacteria is itself expelled by coughing. So folks that have tuberculosis, they, as an example of one of these ways we think about for the way the bacteria can perpetuate itself and perpetuate the disease, it causes coughing, you expel the bacteria, and other people then around you can get it. Let's see. People can have TB infections that are somewhat, what's the term? They're asleep. They are, they've gone into a remission, and they can be reactivated after immunosuppression. HIV infection is one of the mechanisms that can cause immunosuppression, that the virus itself can, and that can result in the TB coming back up. And so other things like smoking and poor health also can result in both a reemergence of the bacteria or a sensitivity to the infection. So you can actually have it and not have an active infection. You have what we think of as a latent infection, and then it can come back up. So there was a term back in the 19th century, early 20th century, known as consumption, which was a disease in which the body was just essentially consuming itself and getting skinnier and skinnier, not an inability to breathe, and this is what happens when you have a long-term TB infection. So tuberculosis, because the bacterium is in the lungs, when coughing occurs, large amounts of the bacteria can be sprayed out. And so consequently, it becomes a public health issue. It is easily transmissible. So TB has been an issue for a very long time, and primarily through good hygiene and through monitoring is how tuberculosis in this country is monitored. People that, we'll get into TB tests in just a moment, but anybody that's in the healthcare biz, anybody that's going to be doing food handling, anybody that's going to be involved in daycare or nursing homes, you all are going to have a TB test to see if you are in fact carrying tuberculosis bacteria. And in general, the trend has been downward. We're starting to see a little bit more of an uptick of late, and the concern about antibiotic-resistant tuberculosis. So one thing is that depending on what part of the world you come from, tuberculosis can be more of a problem, and that begins to manifest itself with immigration and things. So the U.S. doesn't have a vaccine that is used. So we are not preventing infection. What we do is trace it and treat it. In Europe and other parts of the world, they do have a vaccine, but because you have been infected with the vaccine, or you actually have the vaccine injected in you, you begin to develop antibodies, and so it's difficult to actually monitor for the disease that way. So you can't track the spread or the activity of the disease. All right, so let's see. There are a range of different kinds of TB tests, and you probably either have had one or you're going to have one before you start doing clinical rounds and things. And let's see, we got a picture here. All right, so the way that you do a TB test is they're going to take a little, very tiny needle and inject a small amount, a cc or so, of a fluid that is known as tuberculin, which actually was something developed or identified by our friend Robert Koch, who was the German epidemiologist, developed his famous postulates. And tuberculin is a extract of proteins from the tuberculosis bacteria. Nowadays, we've got a purified, very specific set of proteins. And what you're doing is looking for an immune response. If you have recently been exposed to tuberculosis bacteria, and you potentially you're carrying it, you're going to have an immune response in your arm. So you're going to take the tuberculin syringe, just inject it just under the skin. So you're going to just do a subcutaneous injection here, and a little bump, and then you come back. I think within 48 hours. Actually, it has to be between 48 and 72 hours. You come back to your healthcare provider, and they take a little ruler, and they measure the degree of redness and swelling of the lesion. Now, if you've ever had one, you'll know that it generates an itching spot, and I've never seen a positive TB test, but I can tell you that it can be pretty big and still be considered negative. So a positive, I don't want to see. But negative TB test means that you're not generating a particularly strong immune response to the bacteria being injected into you. Now, Koch was hoping that this was actually going to serve as a vaccine. It didn't, but that was the idea. So he was actually, sort of touting his tuberculin as a potential resistance factor, something that you could generate resistance. It doesn't, but it is used as a test. So the TB test is essentially injecting you with a protein extract from the bacterium and looking to see if you have an immune response to it. Drug resistance is a very serious issue in that we've got an increase in multi-drug resistance, and that is a concern because if we don't have antibiotics to treat tuberculosis, we really don't have any kind of treatment for tuberculosis. So it is a serious disease in terms of a population, but it's actually fairly easy to test for. All right, so then we've got this general category of upper respiratory infections that we refer to as pneumonias. And these are typically secondary infections. So you're going to have the flu or some kind of other issue that is going to be a potential immunosuppressant. And then that allows the pneumococcal bacteria it gives them the opportunity to grow in the lungs. And pneumonia is, as I said, kind of a term that is used to describe or is assigned to a set of symptoms. It is not a single organism. So the common bacteria involved are Streptococcus pneumoniae, that's the most common, but Haemophilus influenzae is also found as a pneumonia-causing bacteria. Staph aureus, which is common on the skin and everywhere. This is also able to generate pneumonia. Klebsiella pneumoniae, Pseudomonas aeruginosa. These are other bacteria that, they essentially are opportunistic infections. When you have an immune response, that's when you end up, or an immune suppression, that's when you end up getting these infections. So, pretty high number of cases, about 1% mortality, especially in the old. You can end up with these terms that we refer to as walking pneumonia versus double pneumonia. So, walking pneumonia is referred to as a pneumonia that is actually fairly mild. You begin to see the bacteria and effects in the lungs. You have reduced oxygen carrying capacity, but not so severe. You then have one lobe of the lung growing significantly infected versus double pneumonia, in which you've got both lobes are showing fairly high signs of bacterial infection. So the diagnosis and pathology, you begin to see fluid accumulation in the lungs. This is what's causing the inability to be able to breathe effectively. And normal alveoli are fairly open. With pneumonia, you begin to have the bacteria inside the alveoli, limiting the ability of these cells to pass oxygen back and forth and CO2 from the blood. Necrotizing kinds of effects can lead to general kinds of degradation and sepsis and shock. This is all from the toxins. So different bacteria are going to have different toxins. So pneumonia is not a single kind of disease. Pneumonia is going to have, depending on the bacteria, a variety of different kind of pathologies. So there are primary infections in pneumonia. Mycoplasma pneumoniae is a very weird bacteria that can cause this. These are very, very small bacteria, even for bacteria. They're smaller than standard bacteria. They don't have a cell wall, and they attach to receptors that are at the base of the cilia in the lungs, and this paralyzes and kills the ciliated epithelial cells, which begins to cause necrosis of the bronchi, and you begin to get narrowing of the tubes that are carrying oxygen, the bronchi. And this can lead to chronic bronchitis, bacterial bronchitis. The cilia are responsible for sweeping out dust and things that you might get into your lungs, and these begin to accumulate, and also movement of materials and things around. And so a pretty serious problem. This is an example of a primary infection. It's not one that actually happens it would be characterized as a sudden onset, in that you don't have to have a flu or immunosuppressive event to actually pick up this kind of pneumonia. Now, there are still bacterial diseases that are surprising, that are emerging. Now, you don't remember this, but I was old enough to remember in 1976 that there was this very strange bacteria infection that occurred, I think, in Las Vegas at an American Legion convention. And because it was affecting a bunch of members of the American Legion at this specific site, it's referred to as Legionnaire's disease. The standard kind of epidemiology following Koch's postulates was carried out, in which the causative agent was purified from the people that had the disease, and this is an unidentified bacteria that is now known as Legionella pneumophilia, and it is an intracellular infection by a very common but otherwise or previously unknown bacterium. So what happens is the bacteria actually gets inside the cell through phagocytosis, through the cell's ability to actually pull in a large material, like normally phagocytosis is used in white blood cells to pull in bacteria to break them apart. In this case, we have infectious form of Legionella being brought in, and the bacteria actually start to replicate within the cell, and then they can dump out, and you begin to actually start having cell necrosis and things. So when hosts are not available, the bacteria probably is found on surfaces and forming biofilms. So in the body, it's affecting the phagocytes. These are the white blood cells. And this is going to have an effect on changing the immune system as well. In nature, it's actually found in a variety of different amoeba. So it is an oddball guy, it's an intracellular bacterium. So it ended up being a pretty serious disease, whole body, but it's brought in from the lungs. Turns out that it comes from places like air conditioning vents that aren't being cleaned enough. And so that's what I think in the case of the first disease, it was air conditioning systems that actually had the bacteria in them. All right, so that is covering the respiratory tract, both upper and lower. And we're thinking about things like pneumonia, but also things like Legionnaire's disease and a variety of other kinds of diseases that once they get in, meningitis, they can go to different organs and different parts of the body. So it's not just respiratory infections that we're talking about. It is the upper respiratory and lower respiratory systems' ability to bring things in. That's one of the holes as an entry point. So next, I think we're going to be getting into the GI tract, which will be a lot of fun. Lots of diarrhea for everyone. And that's what I know for now. So I'll talk with you again on Friday. I'll post the PowerPoints here, and I'll see you around.